Good afternoon students, this is your Prasanna Ma'am from NEET World and here we are to discuss our NSEJS biology questions from the paper 2023 NSEJS exam. So far we have completed or we have dealt with the questions of NSEJS biology from 2024 paper and today we are going to begin with 2023. So yes, as we are discussing each question by exercising the concept as well. So today I have brought you two concepts. One is crossbreeding, mutational breeding or cytoplasmic hybridization, and the second question is based on the genetics test cross. So this is a very useful session beta. Look till the end, okay. So the very first question if you have to look into, through the process of crossbreeding or mutation breeding or cytoplasmic hybridization of animals and plants, new improved high yielding varieties or exclusively distinct hybrids are obtained. Which of the following are cytoplasmic hybrids or cybrids? To answer this question, priorly you should have a clear idea on the crossbreeding, mutational breeding and cytoplasmic hybridization. What are the key differences between them? First is crossbreeding. What is crossbreeding, ma? Breeding or hybridization or fertilization between two genetically different variants or genetically different species is called as cross hybridization. Fertilization or crossover between two genetically different variants is called as cross hybridization, which will give you hybrids. What is mutational breeding? Okay, mutational breeding is nothing but by inducing the genetic changes inside the organism, making them more variants is called as mutation breeding. Mutation breeding is nothing but giving out more variants by inducing the genetic changes is called as mutational breeding. Cytoplasmic hybridization is nothing but the nuclear genome of one species crossing with the cytoplasm of the other species or the cell is called as cytoplasmic hybridization, which will result in the hybrids. So of course, to confuse us, he has given us so many terms of cross breeding, mutation breeding and cytoplasmic breeding, but at the end he asked us about cytoplasmic hybrids or hybrids. So the question is only about cytoplasmic hybridization. Once again I will tell you what is cytoplasmic hybridization or how these hybrids or cytoplasmic hybrids are formed. Let us say from one parent, from female parent, let us say the genome or the genetic material is taken like from chloroplast or mitochondria. From chloroplast or mitochondria the genetic material and the cytoplasm is taken, which is crossed with the male parent's genomic DNA is taken. Okay. So the cytoplasm of both the cells are involved, whereas chloroplast genome of one parent and nuclear genome of the other parent is involved in the crossover. Fine, which results in the formation of cytoplasmic hybrids or hybrids, which has the cytoplasmic hybrid or hybrid will have the cytoplasm of the two parents and the genetic material from only the nuclear genome from only one parent. Clear, right, ma? I hope you have understood the question. Once again, for your clear understanding, let me read out the question with explanation. Through the process of crossbreeding, which means crossing between two genetically different variants, or mutational breeding, nothing but increasing the variations by inducing the genetic manipulations or by inducing the genetic changes, creating more variants is called as mutational breeding. Or cytoplasmic hybridization, which is nothing but the chloroplast or mitochondrial genome and cytoplasm of one parent and nuclear genome of the other crossing will give you a hybrid which has cytoplasm of two individuals and genome of only one parent. Of animals and plants, new improved high yielding varieties or exclusively distinct hybrids are observed. Which of the following are hybrids, he is asking. So look at the explanation first, then you will understand clearly. Let us look into all the varieties, all the hybrids and hybrids once and their parents, their variations, then we can easily answer the question. Hybrid are cells or organisms that contain nucleus from one species and cytoplasm from both the parents. Clear right now? It has nucleus from one species and cytoplasm from both the parents. Key point here is, in addition to nuclear genome, they contain mitochondria and mitochondria or chloroplast from other source. Okay, in addition to nuclear genome, they contain mitochondria or chloroplast from other source. Usually it is made via protoplasmic fusion, okay, or protoplast fusion or cell fusion. This protoplast fusion or cell fusion is mediated by a chemical called as PEG. Okay, through this the cell fusion or protoplasmic fusion is mediated, and applications of this include transfer of cytoplasmic traits such as male sterility, disease resistance and improved metabolic capacity. What are the major applications of this? Transfer of cytoplasmic traits as male sterility, disease resistance and improved metabolic capacity. So, this is about what? Hybrid. Okay. Now, let us look into the main varieties of hybrids. Pomato and bromato, these are the varieties which are given in our option, one of the option, that is pomato and bromato. This is the cytoplasmic hybrid or hybrid. By protoplasmic fusion, which is in between Solanum tuberosum, that is potato, and Solanum lycopersicum, that is tomato. So, this is the fusion between Solanum tuberosum and Solanum lycopersicum. So, this is to combine the tomato fruits with potato tubers in one plant, though commercially impractical. This is used for the research. Second variant of this hybrid is in between broccoli and tomato. This is also called as protoplasmic fusion. So, this is an image of pomato. Okay. Now, let us look into the other options as well. Triticale and Fairchild mule. Okay, Fairchild mule. This is one example that is given. Triticale is a hybrid because it is in between two different species. It is a nuclear hybrid. This is not hybrid, okay, which is in between what ma? Wheat and rye. Okay, Triticale is a hybrid, that is fusion in between wheat and rye. Next, Fairchild mule, where animal hybrid is in between male zebra and the female horse. Okay, that is in between male zebra and female horse. This is also purely a nuclear hybrid. Okay, so we have Triticale and Fairchild mule, which are hybrids. Triticale is a hybrid which is formed by the fusion of wheat and rye. Remember that, children. Next, Fairchild mule is the cross in between animal hybrid, that is male zebra and female horse. And then Tigon or Leopon. Tigon and Leopon. Tigon is a variant or a variant species which is formed by the fusion of tiger and lion. Leopon is formed by fusion between leopard and lion. Both are nuclear hybrids only. Tigon and Leopon, both are nuclear hybrids only in animals, and there is no cytoplasmic hybridization over here. There is no cytoplasmic hybridization. Jaya and Ratna rice, both are high yielding rice varieties which are obtained through conventional breeding. These are obtained through what? Conventional breeding, that is nuclear hybridization and selection. This is not cytoplasmic fusion. Okay, this is not by cytoplasmic fusion. So, here if you look into the other three options, one is Triticale, Fairchild mule, these are in between two different variants, hybrids. Tigon and Leopon, this is also a hybrid. Jaya and Ratna, these are formed by what? Conventional breeding. Okay, this is a nuclear hybridization but not cytoplasmic hybrids. So, here you look into this table gives you more clear idea. Whereas hybrid, we already know that hybrid is formed by the fusion of cell fusion or cytoplasmic mixing, not just by the nuclear genome recombination. Hybrid, which is a normal cross breeding, which is formed by combining nuclear DNA from both parents via sexual reproduction. So, here you can see the only hybrid or which is formed by protoplasmic fusion is pomato and bromato. Now, look at this question clearly once again. just a second. Yeah. So, here which of the following are hybrids? Triticale and Fairchild mule, both are hybrids. Okay. Tigon and Leopon, hybrids. Jaya and Ratna varieties of rice formed by conventional breeding. These are also hybrids. The only hybrid which is formed by cytoplasmic fusion, which contains nuclear genome of the one and cytoplasm of both the parents, those are the varieties are Pomato and Promato. Okay. I have given you the explanations for all the 4 options because this stands as a reference for your next exam. Okay. Exercising each option of the question is important, ma. Next, let us look into the next question and then we can directly go for the references. Okay. So, the reference for the previous question that we have dealt just now for NSEJS exam, this is directly there in your biology textbooks about conventional breeding, mutational breeding, cross breeding. Okay. It is clearly there in your 9th and 10th biology sections of improvement in food resources, heredity and evolution. Okay. And in class 11 also the question which is given is linked with cell structure, biotechnology strategies and enhancement in the food production. And in class 12 biology also it is inculcated in the chapter biotechnology principles and processes. If you are referring into the Olympiad specific books, then you can easily get through those topics from biology, Olympiad theory, problems and solutions, biology, and then from Pradeep's biology and then MTG series of NSEJS. From these books you can get the complete concept and objective regarding the previous question. Now, let us look into this question which is based on the test cross. Okay. This question is based on the test cross. So, look at here, phenylthiocarbamide that is PTC has a bitter taste. Non-tasting ability is reported to be due to recessive allele of the taster gene. Okay. So, this non-tasting ability is due to what? Recessive allele. Recessive allele is contributing for non-tasting of the taster gene. That means dominant allele is for taste, recessive allele is non-tasting allele for the taster gene. Understood, ma? Okay, this is non-tasting. In random populations, about 30% people will lack the ability to taste PTC. A non-taster woman is married to a non-taster woman, he is saying. Non-taster woman means it should be homozygous recessive, because small t is only for non-tasting allele in this taster gene. Okay. So, non-tasting woman is married to PTC taster man and they had 3 children. Okay. Look at this properly, ma. So, the female parent is having non-taster gene, that is recessive alleles, and male parent is having taster, that is male can be either homozygous dominant or he can be either heterozygous dominant. The first 2 children are born as non-tasters. What is the probability that their third child would be born as non-taster? They are asking. Okay. So, listen, whenever such questions are given in the complete paragraph, you have to highlight only 3 points, ma. First point is, amongst the gene which they have mentioned, which is the dominant allele and which is the recessive allele, what is it coding for? That is the first point you have to observe. Second point is, amongst male and female parents, amongst male and female, who is having dominant alleles and who is having recessive alleles, that should be identified. And third, only the probability of one child is asked, where you have to be least bothered about the previous 2 children who are non-tasters. The question is nowhere connected to the previous 2 children if they are non-tasters. Just to confuse you, this much big paragraph like question will be asked. The first 2 children being non-tasters has no connection with the problem we are identifying. Okay, so these are the only 3 points that you have to highlight. Look at it once. So this will be the combination. Okay, right ma? Woman is non-taster and males are tasters with dominant homozygous and heterozygous. So let us go for 2 types of crosses. First is in between small t, that is female. Oh, just a second ma. And then male, let us take both of them, capital T in one checkerboard. And capital T, small t also in one. Fine. So this is male and this will be female. Male and female. Okay. So here what do you get? 2 heterozygous alleles. Two heterozygous alleles and 2 homozygous, right? Fine ma. So what do you get over here? First, if you see exactly over here, when 2 homozygous dominant crossed with 2 homozygous recessive, you will get all heterozygous only. Okay. But when this is applied, heterozygous dominant crossed with homozygous recessive, you will get 1:1 ratio. So what is the probability of complete affecting the child? It is just 0.5%. Okay, 0.5% only the chances are there for the child to become a non-taster when heterozygous dominant is crossed with homozygous recessive. So here the answer will be 0.5. Once again for your understanding, I will solve in a different way. So listen here ma. First in the question as they mentioned, woman is married to PTC taster man, non-taster woman, and in the question it is clearly mentioned that non-tasting has the allele small t. So the female is definitely having homozygous recessive allele and the male is having heterozygous dominant or homozygous dominant. Cross with both of them and you will get. First let us do with heterozygous and then with homozygous. Homozygous, so definitely we will get all the alleles as capital T small t only. So there is a no chance here for non-taster kids, but here there is a probability wherein if this is crossed and then, so here you will get capital T small t, capital T small t, small t, small t, small t. So the probability is around, here also it is small t, small t. Okay, it is 1:2, it is 1:1, or you can say half, or you can say 0.5, or you can say half, or you can say 0.5. So the answer is 0.5 over here. So this is how you have to solve any question when it is given for genetics, because mostly the answer lies inside the question itself. Just you have to look at the key points and then solve, that is it. So now the theoretical explanation for this is given, wherein you can see clearly from 0.5% of the chance is there for taster and for non-taster when heterozygous genes are involved. When homozygous genes are involved, homozygous dominant and homozygous recessive, it is impossible for non-taster children. Okay. So this genetics question, we have many more questions as such in 2023 paper, like 2 to 3 questions are there. We will be exercising in our coming classes. So now you see this question, which books you have to refer for the questions like genetics. The books you have to refer, NSEJS reference books include NCERT class 9th and 10th, Hereditary and Evolution, NCERT class 12th Biology, Principles of Inheritance and Variation, Mendelian genetics and probability. So, these are the reference books from NCERT. If you want to take from Olympiad preparation books, then MTG NSEJS Explorer, it has theory and questions, and Pearson MTG Foundation Biology series from class 8 to 10th, it has genetics basics, and then Campbell Biology is the best reference again, Pradeep's Biology or mainly Truman's Elementary Biology, this is the best reference book. Okay. So, these are the reference books and the concepts to be dealt. So, that is all for today's session. In our coming session, we will be exercising few more questions from NSEJS 2023 with clear concept revision. Thank you.